Hello, Anna here, and we're going to go ahead and continue on with our muscle physiology. We're going to be looking at part 2 on the microanatomy. Now you might think that that's histology, but we're actually going to get smaller than our histology. We're going to dig in even deeper into the structure of the fibers, the filaments that are associated with the muscle contraction. So let's go ahead and get started. All right, so let's pull together some notes. If you go to the PowerPoint, you'll see I've already kind of summarized some of this stuff, but the highlights that I really want you to take away is just how huge skeletal cells are, so skeletal muscle cells. So when you think about it, this could be like smooth muscle, and we're going to do kind of like cardiac muscle here, so that's cardiac. And then my skeletal muscle cells are going to be so long that most of the time they don't even fit on the screen. Okay, so really, really big cells. They are also very wide cells, so 10 to 100 micrometers. Okay, so much bigger than other cells. So very long, very wide. Some features that you want to remember that are located in there are things like the glycosomes. All right, and I want you to focus on the word glyco because that is telling you what's in there. So glycogen. Wait, did I just misspell that? G-L-Y, glyco, and nobody can read that now. Glycogen. So glycogen is how animals store their sugars. Okay, so this would be like the carbs that you find in plant cells, but ours are stored in glycogen. So this is really, really important that we have these here, these granules, these glycosomes. Another thing that's important to remember that you've got present is the myoglobin. And I want you to remember that whenever you see myo, it means muscle. Just like whenever you see sarco, it's telling you that you're dealing with muscle. And then globin is indicating that you've got a protein, okay? Now what is this? This is a red pigment. All right, and it's going to hold on to O2 molecules. So it's really helping to store extra O2. So you can kind of imagine how important that would be, okay? Now, like the other muscle cells, we have some special structures in here, like the myofibrils, the T-tubules. We're going to be going over those later, but I need you to remember special organelles that are unique to the muscles, okay? And then we've got the funky names. All right, we've got some name changes that are associated just with muscle cells. All right, I suppose that's probably a little jarring. I went ahead and paused it to write those out because it takes me a long time to write. So sarcoplasmic reticulum, look at the name, sarcoplasmic. Let me change the color. We just look right here. Again, sarco is telling you that it's muscle. But if you change that to endo, you have endoplasmic reticulum. So sarcoplasmic reticulum is endoplasmic reticulum located in muscle cell. Sarcolemma doesn't have a good trick to remember, so you're just going to have to remember that it is the plasma membrane of the cell. And you have to be careful when we get to the nervous system because we're going to have a neurolemma, but it is not the plasma membrane of your neuron. So just be aware of that. And then sarcoplasm, focus on this, plasm, and then sarco. If we change that to cytoplasm, you have cytoplasm. So sarcoplasm is the cytoplasm found within the skeletal muscle cell. All right, let's go ahead and look at some pictures. All right, so right here we've got a picture that's been taken of skeletal muscle cells. And what you're going to notice are the striations. So I've got striations here. So you basically have stripes. So striations are the stripes. And we're going to have a dark layer and a light layer. We can also call them the A bands or the I bands, which is referring to how well light passes through it. So the A bands kind of block the light, so they look darker. The I bands let light go through, so they look lighter. Okay? All right. Let's go ahead and continue looking at different pictures. Actually, no, I want to take some more notes and then we'll look at some more pictures. All right, so let's just review this structure. before we start looking at more pictures. Let's talk about some of our definitions. So when we are looking at a muscle belly, we have to remember that it is continuous. Basically, everything is going to flow from one structure to the next. It's hard to say that there's a discontinuous start and stop. How does it really do this? Connective tissues. All right, the connective tissues, which we are sometimes going to refer to as fascia. Okay, this really helps to maintain that structure. And it's important because it's really important for not allowing tearing. So the muscles are constantly moving, they're contracting, they're under high tensile forces. There's a lot of stretching or tearing, pulling going on. You don't want it to tear. So you're going to use fascia or connective tissues to help prevent that. So the first layer is going to be epimysium, which is going to be a dense, irregular connective tissue proper. I need you to memorize that the epimysium is dense, irregular connective tissue proper, okay? And that's really important because you don't want it tearing. And dense, irregular has collagen fibers going in multiple directions. And when they go in multiple directions, they resist tension or tearing in multiple directions. Then we have the word fascicles. The word fascicle is a fancy word. And this fancy word means a bundle of cells, okay? Or a bundle of fibers. Surrounding each fascicle is another layer of connective tissue proper. And it can be dense irregular or it can be dense regular. It's probably just better to think of it as a fascia or connective tissue proper, a fibrous CT. And that's going to go around all of the fascicles. And then around each cell is a thin smear of areolar CTP. And that is going to be the endomysium. Now remember, areolar also has multidirectional collagen, which means it also resists tension in multiple directions. Okay? Now, let's clear something up really quickly. Language and how we use it. In this case, when we use the word muscle fiber, it means the exact same thing as muscle cell. This is not true with the neuron. Okay? So be really careful when we get to the next chapter and don't get these concepts mixed up. So muscle fiber is the exact same thing as muscle cell. And in fact, it's referring to a single muscle cell. A single muscle fiber is a single muscle cell. Okay? Now, when the muscle cells contract, they pull on the adjacent connective tissues, which helps to move everything around it. And the connective tissues also help to prevent the muscle cells from overdoing it and tearing. Now, let's look at a picture. All right, we're going to work out from big to little. So right here, I've got a bone. Here is my tendon. All right, the tendon is not the same thing as muscle, and it's not the same thing as a ligament. We'll talk more about it later. But be aware that they're not exactly the same thing. Now, I want you to look at this entire thing here. The entire block of red tissue is the belly. Everything that's in it. All right? That is your muscle belly. Now, notice this white film that wraps all the way around it. That is your epimysium. And remember, that's going to be typically a dense irregular CTP. Okay? Now, in here, within this, I've got these things that look like people have taken bundles of grains and wrapped them up together. All right, each bundle is called a fascicle. So here we've pulled out one bundle so that you could see it better. Now, wrapped around the bundle is another white film, and that white film is called paramecium, okay, which is also a CTP, all right, connective tissue proper. Now, within the epimecium, I have cells. So there's a cell, there's a cell, there's a cell, and these can also be called fibers. A single muscle cell, a single muscle fiber. Around each one of these is, again, white film. That is the endomecium. Then within it, the cell, I have these round things here. These round things are called myofibrils, and they are the organelle for contraction. But I can also see here and here the nucleus. And I can see here and here and here and here the light bands and the dark bands for those striations. Okay? All right. Let's talk a little bit more about this myofibril thing. All right. That myofibril. So I need you to memorize that it is the organelle for contraction, that it's its definition, that is what it does. Okay, that you have to have memorized. This is the organelle used for contraction within muscle cells. Now, within a single muscle cell, there will be hundreds to thousands of myofibrils, and they are packed tight within the cell. All right, they're packed so tight you can hardly see anything else. So they're really, really, really dense, really, really important. Okay, the organelle itself, if you break it into pieces, it is a string of sarcomeres, and I'm going to explain that a little bit more later. I just want to put it all in the notes in one place for right now. Now, you need to be careful in that you don't confuse a fiber with a myofibril with a myofilament. So we're going to write that down. Fiber, myofibril, myofilament. Fibers are big. Myofibrils are smaller, and they are within the fiber. Myofilaments are teeny tiny, and they are found within the myofibril. So you need to keep that straight. Now, I've mentioned that myofilament. Now, the myofilament is basically proteins. And we got two really major ones that we're going to talk more about, actin and myosin. All right, we're going to get into those in just a minute. We've also got, and in fact, let me clarify. I don't have room. Contractile proteins, so we're just going to make that one. They also have regulatory proteins. And the two major ones are going to be troponin and tropomyosin. Okay. Then there's one more protein we're going to talk about, and it is an accessory protein, and it is called dystrophin. All right, so find some way to kind of write that out and start memorizing them. I would kind of like pull together all of my slides and try to put all of this information into a concept map or into a table to help you with memorization. Okay, now we're going to go through, and I'm going to quickly, you know what, I think what I'm going to do is I'm going to start talking about pictures. What I want you to do is review the PowerPoint. I've got two slides in there on the myofibril structure, and I basically give you the definitions. Review slides 22 and 23 for definitions. But I'd rather go ahead and start talking about the pictures than trying to read off a bunch of definitions. All right, so let's go on and look at pictures now. All right, here in this picture, I have a single cell, which can also be called a single fiber. If we kind of start over here, right there, what you are seeing is the sarcolemma. All right, and remember, the sarcolemma is your plasma membrane, and right there they've got the label. Okay, so this is going to wrap around the whole thing. Here I can see a cute little nucleus. I can see another one there. I can see another one there. Okay, now remember, within the cell you have the cytoplasm, and the cytoplasm is basically going to be cytosol plus the organelles. So there's a little bit of cytosol, which is that liquid background, and then there's a whole bunch of other stuff within it. And within it is the nuclei. All right, so just right here, like I did the nuclei, okay? But you've also got mitochondria, and you're going to have quite a few mitochondria because you need to be making ATP energy for the entire cell all along the length of it. And I can see more, whoops, here, here, here. So lots and lots of mitochondria. All right, now within that sarcoplasm, we also have the myofibrils. So there's one, here's a myofibril. There's one, another. So these are all myofibrils. Now, when you look at this, you will see the light band and the dark band, and you can see right here they've got it labeled, okay? You're gonna start seeing, you know, you've got the striations, you've got this nice stripe, and you're gonna start appreciating now the Z disc here going down the bands, okay? And you can start getting an appreciation for how on either side of the Z disc we have this blue and purple line. So this is where your T tubules and sarcoplasmic reticulum are located, and they are literally wrapping around all of the myofibrils. And you can see over here where they've been cut. Okay, so it's literally wrapping all the way around all of these little doohickeys. All right, so let's look at another version of this and start going in even deeper. Okay, so here on this particular picture, we've got a cut cell. So we've only got a little segment of it. They've pulled back right here the sarcolemma so that you can see that, and then you can see the nucleus here and another nucleus there. It's hard to tell, but there are mitochondria in here as well, okay? You can see the striations very nicely for the I band and the A band. Now what they've done is they've taken a single myofibril and they've pulled it out like a straw. And then they're going to bring it down here and they're going to blow it up so that you can see the dark, light, dark, light, dark again. And then you can see the Z disc here and a Z disc there. Now we're going to take this little section and we're going to blow it up and bring it down here. Now we've got a really important thing. Sarcomere. So from here to here, this is called a sarcomere. So you can see in this picture there are 1, 2, 3 sarcomeres forming a chain of sarcomeres. And what's important is that when an organelle contracts, it contracts from one Z disc towards the center of the sarcomere. So that a contraction is the accumulation of a bunch of tiny contractions towards the center. Now we're going to blow up the sarcomere so that you can see the structure a little bit better. So you've got the Z disc here and the Z disc here. And then what you've got are, what have they done here? I've lost some of my arrows when it moved it from one program to the next. So each one of these blue lines is called an actin protein. And we call these lines also thin filaments. And then right here, the red lines, those are all myosin proteins, and we call these thick filaments. And then we'll just continue using red, and we didn't really mention this on the previous slide, but we also have elastic proteins at each Z disc, which help with recoil after the contraction is done. Okay, so let's blow this up even bigger and focus in on it. All right, now we're looking at the sarcomere done big. Okay, we've got again a Z disc and a Z disc. Within the Z disc, we have the elastic filaments, which are made out of a protein called titin. And when the contraction is done, these help to recoil and bring these things back into a resting state a little bit. Okay, in blue, we have, oh, I don't like that color. In blue, we have actin, which is also called a thin filament. And in pink, we have the myosin, which is also called a thick filament. Now, where the thick filament exists, it blocks light. So the thick filament is responsible for the way the A band looks. The thin filament is where you just have actin and the elastic proteins. Because you don't have the thick filament, less light is blocked, and so it's a lighter color. Okay, so what do you need to memorize? You need to memorize that actin goes with thin filament. You need to memorize that myosin goes with thick filament. You need to memorize the Z disc, A band, I band, and to a lesser extent the elastic. I would like you to draw out this picture, and I would like you to label it and be able to talk about the functions of these different structures. Okay? So this is a really, really important picture. It's a really, really important diagram to understand and to be able to visualize without effort. Okay, so do it a couple of times. Now let's look at what happens during a contraction. All right, so we're looking at 3 different muscle cells at different states of contraction. And I'm sorry, this did not import well with the words when it was converted to a PDF, so I wrote in blue off to the sides. So up here, this is the muscle cell, the sarcomere, when it is in a relaxed state. I have overlap between my thick filament and my thin filament, but there is a gap where they don't overlap. You can see how much this is not super tightened up, the elastic fibers. Okay, so this is relaxed. Now here we're going to look at partial contraction. And what has happened is that this pulled towards the center, and you can see the Z discs are closer together. You can also see right here that the gap between the thin filament and the thick filament is much less. So it's pulling towards the center. Now over here at the full contraction, so again, you're pulling towards the center so that the Z discs are now even closer together. And now if you look right here, you can see that the thin filaments are even overlapping. So very tight contraction right there. Now remember, there would be Z discs on either sides of this. So this one would be contracting in that direction. So that when you have a very long chain of sarcomeres, full contraction of the entire myofibril is the accumulation of lots of tiny contractions. Or lots of tiny movements at each sarcomere. Okay? All right, let's take a peek at that dystrophin. All right, so I want you to focus in first on the sarcolemma, okay? And then this is outside the cell, and this is inside the cell. Now, this is representing the myofibril. Now, if the myofibrils are within the cells and they contract, why does the rest of the cell contract around it? How is that happening? Why don't the myofibrils just get seriously short inside the cell and nothing else moves? Well, the reason this affects the entire cell, including the sarcolemma, is because of the dystrophin. The dystrophin is a little tube of connector protein, and it goes from this thin filament of your sarcomere and hooks into some other proteins that go through the sarcolemma and also anchors into the endomysium, so that literally you tie it together so that when this part contracts, this gets dragged as well, so that the entire cell shortens, not just the myofibrils within it. And this is a very important, clever little mechanism. And when you may have heard of people with muscular dystrophy, so muscular dystrophy affects that protein. And without that protein, the cells contract, but the rest of the structures around them don't move. So it's an ineffective contraction. Okay? All right, so I want you to remember that about dystrophin. That'll probably pop up on the exam because I think it's seriously cool. So it's got serious cool factor. It's a test question. All right, let's summarize our structures from small to big. All right. One of the things that was tricky for me was to kind of visualize the relationship between all these words and to be able to see how they all interrelate. So one thing that helps me is to kind of think about size and how they're like folding within each other. So I want you to think of your actin and your myosin, okay? Those come together. Those are myofilaments. And then those myofilaments, if you string them together, you get these cute little sarcomeres. And then the sarcomeres, if you chain them together, you get a myofibril. All right, and remember that myofibril is the organelle itself, okay? Now, if you pack a bunch of myofibrils together and you wrap them all up, you get a muscle fiber, which can also be called a muscle cell, okay? Now, the muscle cell has wrapped around it its sarcolemma. And remember, the sarcolemma is its plasma membrane. And the plasma membrane, we're going to link with dystrophin, all right, to the connective tissues around it. So in this case, we're going to link it to the endomysium. Okay? Now, if you bundle a bunch of fibers together, you will get a fascicle. And surrounding the fascicle, you will get the paramecium. And if you bundle a bunch of these, if you bundle a bunch of these fascicles, you will get a muscle belly. And surrounding the muscle belly, you will get epimysium. Okay? And then in my notes, I could go in and put things like dense irregular CTP. I could put in here a reeler. I can add more notes. So here I've just kind of made a visual diagram that helps me remember how these things are fitting together and structured. So I want you to come up with your own method and to do the same thing some way, whatever works for you. All right, so that was the end of this part. So I'm going to go ahead and stop here and you can take a break.